Hello students, welcome to EPG Pathshala. Myself Dr. Bhashyati Banerjee from Gautam Buddha University, Greater Noida. In this module, I will guide you through the module titled Classification of Genomics with special focus on functional genomics. This is a module under the paper Recombinant DNA Technology and Genetic Engineering of subject Biotechnology. So students, in this module, we aim to give you an understanding about the fundamentals of genomics and about the conceptual difference between the two disciplines, genetics and genomics. In this module, we also introduce you to the phenomenal Omics Revolution as we guide you through a just timeline of progression from genetics era to genomics era. Thereafter, this module focuses on classification of genomics into classical and advanced genomics, which is then classified into structural and functional genomics. In the concluding segment, this module gives you a detailed overview about the various techniques and applications of functional genomics. Before proceeding on to a detailed discussion, let us begin with the basics. So, in the opening segment of this lecture, we introduce the topic beginning with the definition. In the simplest terms, genomics may be defined as the study of genes and their functions. Let us now take a look at some official scientific definitions of genomics. In more technical terms, genomics is defined as that branch of molecular biology which is concerned with structure, function, evolution, and mapping of genomes. As it was documented way back in 2002 in the report of World Health Organization, genomics was then referred to as the study of genes and their functions, and of course including the related techniques. But even then, what does genomics actually imply? Genomics implies a collection of tools and techniques and methods thereof which collectively aim to understand the structure and the function of genome as a whole and thereupon integrate them to include in mapping of genes and sequencing of DNA. In a nutshell, genomics examines the molecular mechanisms and the interplay of genetic and environmental factors, in particular with special reference to human diseases. In order to understand the difference between genetics and genomics, we first need to know the difference between the gene and the genome. The primary difference between genetics and genomics is that the genetics includes study of functional characterization of individual genes of an organism, while genomics as a subject addresses the structural and functional interrelationship among all genes of an organism collectively. It also includes all the tools and techniques which are required to analyze the combined influence of all its genes on the growth and development of the organism. So now in this segment of the module, we introduce the concept of omics and the Omics Revolution. We begin with a timeline study of progression from genetics era to genomics era, and then we highlight a few important omics techniques. Although DNA was first isolated in 1869, it was not until more than a century later that the first genome was sequenced, making genomics a relatively new discipline. Genomics, as we know it today, truly began in 1970s, although there were several significant milestones that shaped the field in the preceding century. The term genomics was coined way back by a geneticist, Thomas Roderick, in 1986. He was a geneticist in Jackson Laboratory, and in a meeting which was conducted about human genome, he coined this term, which later on was adopted by all the biotechnologists thereafter, and it became a household term. The birth of the term genomics, as documented by the editorial board itself of the journal Genomics, is as follows. To quote them, "For the newly developing discipline of mapping or sequencing including the analysis of the information, we have adopted the term genomics. We are indebted to T.H. Roderick of Jackson Laboratory, Maine, for suggesting the term. The new discipline here is born from a marriage of molecular and cell biology with classical genetics and is fostered by computational science. However, an ambitious project, the Human Genome Project, was launched in 1990 with the aim to sequence the entire human genome. What I mean here is the entire 3 billion letters contained in human genome. However, it was only in 1999 that the first human chromosome, chromosome number 22, was completely sequenced. In between, sequencing of the first bacterial genome, yeast genome, and the genome of C. elegans was completed. Dolly, the first cloned ship, was also born during this time. Another landmark year was 1997, when the E. coli Genome Project was launched, and at the completion of the project, 4.7 megabase pairs of E. coli genome was completely sequenced and shown to contain 4,500 genes. The first full mammalian genome, that of a mouse, was sequenced and published by the International Mouse Genome Sequencing Consortium in 2002. Later, in 2003, the flagship Human Genome Project was finally accomplished, and it confirmed that humans have around 20,000 to 25,000 genes. The International HapMap project was then launched to create a catalogue of common variations in human genome. In 2007, with the advent of next-generation sequencing and high-throughput sequencing technologies, another significant breakthrough was achieved. It was here that in the field of genomics, 70-fold increase in output of DNA sequencing in 1 year was achieved with the help of next-gen sequencing. All these developments together led to the launch of the 1000 Genes Project in 2008, which aimed to sequence genomes of large population group of 2,500. The result of this project was published in 2010. As the research in biotechnology progressed from genetics to genomics, several distinct components or entities were identified within a cell. To name some important entities, genome was described as the haploid set of chromosomes in a gamete or microorganism or in each cell of a multicellular organism. In other words, genome refers to the complete set of genes or genetic material present in a cell or an organism. Genome produces transcriptome, the sum total of all messenger RNA, sometimes referring to the entire RNA transcripts expressed from the genes of an organism under given circumstances. Transcriptome translates into proteome, the entire component or protein complement of a cell which is or can be expressed in a cell, tissue or organism under given circumstances. Metabolome was a term coined to refer to the complete set of small molecules found within a biological sample or a tissue sample. A relatively new term is ionome, which is defined as the mineral, nutrient, and trace element composition of an organism that represents the inorganic component of the cellular and organismal systems. Now there is a huge uproar about omics everywhere. This era of genomics can be considered as an era of omics revolution. But what does omics signify? Omics means, in a very simple term, omics refers to the application of techniques and profiling and analysis of the data obtained thereof in order to characterize completely the various aspects of a particular entity. In context of cellular biology or biotechnology. Omics refers to application of techniques of different disciplines included in biotechnology in order to characterize the different entities which we have just discussed previously. Thus, genomics refers to the application of techniques of genetics and molecular biology to genetic mapping and DNA sequencing of sets of genes or complete genomes of selected organisms. Genome produces transcriptome, and transcriptomics is the study of entire transcriptome produced by the genome under specific circumstances. Transcriptome translates into proteome, and proteomics is the application of techniques of molecular biology, biochemistry, and genetics for complete structural, functional, and interaction profiling of proteins produced by the genes of a particular cell, tissue, or organism. Metabolomics is the scientific study of chemical processes involving metabolites. Ionomics, the analysis and profiling of ionome, requires the application of high-throughput elemental analysis technologies and their integration with both bioinformatics tools and genetics tools. Now, you must remember that all the omics techniques include organizing the database and designing practical applications of the resultant database, as in case of medicine or biology. So, now that we have introduced omics substantially, let us now specifically start decoding and deciphering the genomics itself. In this segment of the module, we will discuss about the early developments in the field of genomics, which today is referred to as the classical genomics. We shall also give you an understanding of development of various new sub-disciplines of genomics and how these new additions led to the emergence of modern genomics. Genomics emerged as an epitome of multidisciplinary science. It emerged by the convergence of many disciplines such as genetics, molecular biology, biochemistry, statistics, and computer sciences. It stemmed from the discovery of gene linkage, that is, many genes present on a single chromosome, which led to the construction of linkage map. Towards mid to late 20th century, it marked the advent of classical genomics era. Classical genomics played a critical role in bridging the gaps between genetics and other branches of genomics. Though it might have taken a backseat in the light of simultaneous rapid advancements in many other disciplines, but genomics was revived and rejuvenated in full flare in late 20th century with the spectacular development of molecular markers, followed by the launch of Human Genome Project and Plant Genome Projects. Classical genomics was based on the integration of several traditional as well as modern disciplines. It included the tenets of Mendelian genetics, cytogenetics, molecular genetics, quantitative genetics, population genetics, and of course, genetic mapping. It mainly utilized applying the fundamental tools such as genetic markers, linkage analysis, gene ordering, multi-point analysis, genetic mapping, and quantitative trait locus mapping or QTL mapping. As the data from classical genomics started pouring in at a large scale, it became imperative for the scientists to develop at par in silico processing tools, and this led to the emergence of genome informatics, which utilized genome databases, sequence comparison, data communication, and automation in order to achieve in silico genome and data analysis. By the first decade of 21st century, there was substantial progress in the field of genomics and addition of many new branches, tools, and techniques. It was time to classify genomics into many distinct sub-disciplines. To begin with, genomics was initially classified into classical genomics and genome informatics. Eventually, classical genomics made way for structural genomics, functional genomics, and mutational genomics, while genome informatics evolved into bioinformatics and computational genomics. Genomics, as we already know, is the study of genes and their functions and aims to understand the structure of the genome, including mapping of the genes and sequencing of DNA, and most importantly, the molecular mechanisms and interplay of genetic and environmental factors in disease. Thus, distinct disciplines of genomics were established with distinct objectives. Functional genomics was established with the aim to deal with characterization of genes and their mRNA and protein products. Structural genomics was established to deal with the dissection of architectural features of genes and chromosomes. Comparative genomics focuses on the evolutionary relationships between the genes and proteins of different species. Mutational genomics involves profiling the mutagenesis, both natural and induced, and their impact on the genotype and phenotype, especially in context of carcinogenesis. Epigenomics, also known as epigenetics, aims to analyze and understand the epigenetic profile of an organism, including the DNA methylation patterns, imprinting, and DNA packaging. Pharmacogenomics includes study of new biological targets and new ways to design drugs and vaccines. The main objective of structural genomics was to achieve high throughput determination of protein structure of every protein encoded by the genome by using a combination of experimental and modeling approaches. On the other hand, the primary objective of functional genomics was to analyze the function of genes and the proteins they produce, and thereby to expand and correlate genomic and proteomic knowledge to explain the dynamic properties of an organism at cellular level and to understand the relationship between an organism's genome and its phenotype. The structure-function correlation can be immensely beneficial in the practice of manipulating genes and DNA segments of a species for the purpose of targeted genome engineering. Now, in the next subsection, we will give you a brief understanding of structural genomics in particular. The main objective here is to give you an idea of the features and aspects which distinguish the two fields, structural genomics and functional genomics. So let us first take a quick look at the scope, techniques, and technologies used to study structural genomics and then summarize their applications. The principal difference between structural genomics and traditional structural prediction is that structural genomics attempts to determine the structure of every protein encoded by the genome, while traditional structural prediction focuses on one particular protein only. Hence, techniques used in structural genomics are aimed to cover entire genome proteome context. They may be classified into de novo methods and modeling-based methods such as cloning and expression of every ORF as opposed to ab initio modeling, sequence-based modeling, and threading. The main aim of the structural genomics was to study the structure of entire genome by utilizing both experimental and computational techniques. And the primary advantage here is that genomic modeling based approach is not limited by experimental constraints. Knowledge about the composition of genes is always an additional advantage when attempting to understand its function and also helps to design strategies for manipulation of genome to ultimately improve health. Here we summarize the applications of structural genomics, and it's very clear that it has got potential implications for drug discovery, protein engineering, and finally aims to identify novel protein folds and three-dimensional structure. The ever-increasing protein structure database facilitates improved structure determination methodology by more precise homology analysis of unknown or newly discovered proteins. Students, we have now arrived at the most important segment of our module. In this segment, we give you a detailed overview of functional genomics in particular. The main objective here is to give you a brief idea of scope and applications of functional genomics. Thereafter, we guide you through various tools and technologies and sub-classifications of functional genomics. The focus of functional genomics is on a genome-wide analysis of functional role of the genes, rather than focusing on individual genes as in case of classical genomics. It includes studying the patterns of gene expression, interaction at the genome level, and measuring the expression of levels of RNA and proteins produced. It aims to assay and analyze the dynamic aspects of genome and the genes, such as transcription, translation, and protein-protein interactions. Modern biomedical research involves a merger of molecular medicine and functional genomics with an aim to revolutionize the impact of the future therapeutics and treatment methods of human genetic diseases. Since functional genomics covers a huge area of study, hence the tools and techniques are also widespread and may be classified into various categories. There could be DNA-based tools, which include genome profiling techniques, RNA-based tools including transcriptome profiling techniques, protein-based tools which aim to study the protein-protein interactions. Some tools and techniques of functional genomics also include disruption-based studies which aim to disrupt the gene in order to analyze its function. Some techniques used functional annotation-based studies. And finally, there is the integrative approach, which aims to integrate the bioinformatics tools and non-bioinformatics tools in order to produce constructive databases. Genome profiling methods are centered around DNA sequencing and profiling. Systematic pairwise deletion of genes or inhibition of gene expression can be used to identify genes with related functions, even if they do not interact physically. Epistasis refers to the fact that effects for two different gene knockouts may not always be additive. That is, the phenotype that results when two genes are inhibited may be different from the sum of effects of single knockouts. Genome profiling techniques also formed the basis of ambitious projects like Human Genome Project and the Project ENCODE. Transcriptome profiling methods are centered around quantitative assay and sequencing of various types of RNA transcripts which are produced in a cell. cDNA microarray is a popular method to assay the gene expression where differential RNA expression is assayed at microscale through cDNA hybridization. Serial analysis of gene expression or SAGE technique is another very efficient and painless gene expression profiling method. It is a transcriptomic technique used by molecular biologists that allows simultaneous, rapid, detailed analysis of thousands of transcripts in a cell. Here we depict a schematic flow diagram and different types of SAGE that are commonly used. MicroRNA sequencing is another very popularly used transcriptome profiling tool. Here, the image on the left is a schematic diagram of microRNA sequencing protocol, where microRNA are sequenced by ligating RNA adapters to each end of the mature microRNA, followed by reverse transcription and PCR. in other terms, RT-PCR. The image on the right-hand side shows steps in analyzing the compatibility of sequenced RNAs with microRNA biogenesis. The protein-based methods employed in functional genomics mainly utilize the protein-protein interactions. Most popular ones are yeast-2 hybrid system and the affinity purification clubbed with mass spectroscopy technique or APMS technique. Yeast-2 hybrid assay is employed to check the interactions between two proteins known as the bait and the prey. When the fusion proteins are produced, the bait part of the fusion protein interacts with the prey part of the second, and this leads to transcription of the reporter gene. The workflow of APMS for high throughput screening is depicted in the image on the right. Cells that express an epitope tagged form of the protein of interest are lysed to extract the protein complexes. Then affinity interaction is used to isolate and purify the protein complexes with the help of a tag-specific antibody. Following this, separation either by gel or elution, which is digested to generate peptide fragments that can be analyzed by mass spectroscopy to identify co-complex protein, is performed. Disruption of a particular gene or a part thereof is another sure-shot method to identify and characterize the functional aspects of that particular gene. Disruption of gene function may be achieved by several strategies such as mutagenesis or gene silencing. Gene silencing implies the technique which is aimed to reduce or eliminate the production of a protein from its corresponding gene. Small interfering RNA, short hairpin RNA, microRNA, etc. are widely used to achieve gene silencing. CRISPR-I is a prominent example of this technique. Mutagenesis is another strategy to eliminate a gene function. It may be induced at several levels. It could be directed to a specific nucleotide, as in site-directed mutagenesis, to a transposon, or could be aimed to remove or knock out a complete gene as a whole. Now, we move on to the functional annotation-based methods. Genome is in itself a pretty big entity. In order to make sense out of it, we need to break the genome into smaller sized fragments. In order to ensure proper information extraction from the data produced from these fragments, we need to annotate them precisely. The image here shows a generalized flowchart of genome annotation based on statistical gene prediction, which uses methods like GeneMark or Glimmer to predict protein coding genes. Rosetta Stone approach is a popular method for de novo protein structure prediction. The highlights of protein folding and de novo protein designing for biotechnological applications include interplay between accurate protein structure prediction and successful de novo protein designing. It finds widespread applications in therapeutics, as biocatalysts, and in nanomaterial designing. Since we are talking about functional genomics, the immense contribution of bioinformatics must be separately highlighted. Bioinformatics has played a crucial role in the development of present functional genomics. The techniques of functional genomics produce a lot of high throughput data, which cannot be comprehended by unaided human brain. Computational tools are thus required for better understanding and analysis of data in order to deduce a constructive correlation. Conservationists, anthropologists, social scientists have all made use of the data obtained from functional genomics to evaluate several key factors. Conservation biology, preserving genetic diversity and gene pool, determining evolutionary lineages are some of the important functional genomics applications in social sciences. Students, that's all in this module for now. Hope you had a good insight about functional genomics. So students, now let us summarize what we have learned in this module. The knowledge and understanding acquired from genomic research can be applied in a number of different settings, including medicine, biotechnology, and social sciences. Although the applications of genomics have already found a place in many areas, there is potential for study to revolutionize fields involving human and other life on Earth. Recent developments in genomic research, especially in functional genomic research, have opened up immense new research avenues for medical, biotechnology, and biomedical research. This has provided a much deeper insight of genomics and the role of genes in causing or preventing diseases. Notable applications of genomic research in medical and biomedical science are summarized as follows. Designing of personalized precision medicines, oral immunization through plants and synthetic food, designing of malarial vaccines, and screening of thalassemia are some of the important medical applications. On the other hand, important biotechnology applications of functional genomics include analysis of profiling of NGS data, synthetic biology applications, and identifying novel proteins and associated genes and deciphering the functions thereof. Further, conservationists, anthropologists, social scientists, and various other groups have made use of data obtained from functional genomics to evaluate several key factors. Functional genomics finds wide applications in conservation biology, in preserving genetic diversity and gene pool, in determining evolutionary lineages, and in several other anthropological studies. So students, that's all about classification and introduction to functional genomics. Hope you enjoyed the module.